Good morning, everyone. Welcome to Neuroccino. Two people pre-registered that they want to present today. So I just quickly repeat the rules of, sorry, I'm really blurry. I'm going to clean my camera once someone else is presenting. But the rules of the game is briefly present the paper, something exciting from last week ideally, or a historical paper, or a book that's a new category, and tell us why you're excited about it, what you understood about the paper, and we have a discussion about it. And because I'm super smudged on my screen, I'm going to hand over straight to three papers. Leah, do you want to start?
Yes, I'm gonna share the screen.
I'm just gonna clean my lens, but I'll be here.
Okay, can you hear me? See the screen? Everything? Yes. Okay. So this paper is not from last in 2020. I found it really interesting, like a new perspective that I never think about. So I wanted to share with you this piece of information and discuss about it. So this is a paper by Denis Levian, and is about a relativistic pseudo-diffusion framework in the brain. So we are here discussing a new concept, so that we are not only looking to how brain regions are connected, but also the time relation that we have between different events that can happen in your brain. These events usually are brain activation. So the velocity of by which electric impulse is propagating through the brain is not like infinite, or there are some limits. For example, here he's reported that for short amyelinated axon is 0.3 meters per second, or it can go up to 100 meters per second. And so there is a velocity, and if there is a velocity, we can see this event with different perspective. So I don't know if someone is familiar with this relativistic framework, but for example, if the typical example, if it's I'm like on a boat that is like leaving moving in the sea. If I see an event in the boat, I see the events that are like outside the boat. I see that in other velocity. You can like in a train. If you are on the train, you see something dropping, you see that in a way, or if it's outside and you are moving, it's different. So it's all about perspective, about activation, how activation in different brain nodes are connected between each other. And for describing these events, in this paper, they talk about brain lines. So brain lines is like a sequence of events that can be interconnected. So there is, it's not just a network, but here there is a sequence. There is the past, the present, and the future. So there is a movement through activation. So we are talking about velocity. And so here we are talking about brain lines, and to do that, in this paper, he's used the Minkowski diagram that is a relativistic typical representation of events. So you have in one coordinate the time, and here you have the space in x, y, and the brain lines events, and these are the cones of events, and if the brain lines is passing through different cones, these are all the possibility that you can have that one event is interconnected with the other event. I hope that this explanation is not too messy, but and here the velocity is the slope, so the angle of these brain lines, and so the idea is that you are discussing about a new coordinate, so not only the space. So if you have a connectome, we are not just talking about how the structure is of the connectome, but we are also talking about the propagation by Einstein. We talk about the gravity and how the space-time is modeled. So there are curvature that are is the gravity modifications given by the nodes that are the nodes of the connectome. So we imagine the brain as a connectome, and the energy of each node, that imagine that like an hub or with a lot of activation, this energy shape this space, and so all the connections. So for example, a typical network measure that we usually talk about is the shortest path. So here we have to imagine in this new framework with also time, not just the shortest connection path has the shortest, but also like the fastest. So we are also discussing about time, and to do that, we in the paper are proposed new modeling of these, and I'm not going to the details, but for example, another discussion is if there is diffusivity between axons, and this diffusivity, as we usually have seen this equation that is the Brownian motion. So here we are supposing that particles can have infinite velocities. This is actually in conflict with the reality, where the velocity are limited to the structure of the connectome. So this is what is arguing this paper. And so to improve this framework, here you have the Lorentz factor gamma. This is a really relativistic description where you are imposing that there is a limit in the propagation of, so a velocity limit that in the relativistic framework described by Einstein, it was the light velocity limit. Here is the limit given by the connectome. And to describe all these, implement or these hypotheses like mathematical modeling, here there are some simulations where you actually have a grid, and you see and you recreate this event chain of the brain between the idea here is how we are modeling the brain in this framework. You have a connectome then you have connections between different nodes that are so that you can have in your brain, and the connection is not given by this new variant. And this is modeling your space, so give like slopes, so in the activation that are also given by the time variable. And then here are discussed some possible application of this new framework. So for example, there was a nice description where it was here about these connectome disorders. So here, if we are also considering time, like this connection is not just that there is not a connection between two brain regions, but maybe this connection has a synchronous time, like a chronology that can give problems. And for example, here in schizophrenia, like are described the auditory hallucinations, where maybe it's not that there is not a connection between the brain regions activated, but maybe these connections has some timing that can make problems. So for example, like some of your internal thoughts that can happen in the brain are recognized before by the auditory cortex, before then, for example, the frontal brain regions. So it seems that these voices are from outside and not from like your internal thoughts, and that make the trick maybe of hallucination. And or here is also described this the Rubin's figure, where it's shown a figure and it's asked if what you see. You can answer if you see a face, faces, or a vase. And his is hypothesized that a different chain of events, so a different synchronous activation, like no, it's not synchronous, but a timing of activation in this network can bring you to a different answer. And so I think and also like it's also described like when you have a deja vu. So some of it seems that something that you are experiencing in reality, you already have a memory of it, and it seems that the memory is bring back in the presence. So this is another example where like the timing between brain activity can make you like a different sensation of what you are experiencing. So this is what I understood from this paper. I hope that it was not too messy to explain about. And I want also to ask you if what you think, have you ever think about the timing in the brain? I was also wondering if there are some like measures, like empirical measure that we can have, maybe AG or I don't know, some different measures, because it seems that like with fMRI, maybe, I don't know, are we able to have this timing of activations? Maybe it's too fast. Can we, I don't know, can we use these like also in some applications in what we are studying in pathologies, or just a new framework that we can think about? Actually, I never heard about this relativistic framework in the brain, that this is what also is described in the introduction. And so, yes, this was the paper. If you have any question for us.
Thank you, Leah. Right, I fixed my camera, I think, so that should be okay now. Any questions in the room? Yes, go ahead.
Yeah, hi, ma'am.
Hi.
Thank you for the lovely paper. can I get the link for this paper if it's possible? And my
Is
question.
in the box
Okay.
in Zoom
Oh,
at the
okay,
beginning.
So
okay.
sorry to interrupt briefly. For the
Yeah.
links, they're usually in the chat, and then when we edit the video on YouTube, you have the links in the
Okay.
description
Okay.
box and on Twitter as well, and Facebook,
Right.
wherever you follow us.
Right. My question was about the space-time being a mathematical construct, right? In physics, uh, so how it would be translated to a physical construct in the brain in terms of connectomes. Uh, I was wondering whether uh, uh, the limitation of the space in the brain the uh, and the time frame where each node or the each network uh, uh, respond to the space dimension in terms of perception uh, could be could be same as or could be interpreted as space-time dimension, what it is uh, indicated in relativistic features in physics. Because it's a complex idea in physics, right? The space-time dimension being one. So how the brain respond to the mathematical construct could be different to what uh, our perception would allow. What do you think about it?
Yes, I think that of course there are like similarities with the typical relativistic framework. So where if you put together space and time, and there is the limit of velocity, that could happen that time is also like a variable. So it's not just fixed, but the perception of time can change between the perspective where we are looking these. And in the brain, of course, the energy is like the field is different, because here he's hypothesized that the field is given by the nodes activation, like energy. So to me, the interpretation, like the hallucination explanation, give me like a key to maybe see an application, so a perspective of this brain relativistic framework. So like everything like maybe in timing. So I see this as an application of timing, but I see what you mean. That is not just timing, but is that time is can be like different. So an event can be like shorter from one perspective or longer from another perspective. And I don't know this how can deal with like synapses, because this is not just in the brain activation that you have the propagation. Then you have also like other things going on. So maybe like a variation then in like neurotransmitters, like in the creation of like other things that are given to are needed for brain activation and like neurons activity. Maybe these different timing, so also time perception, that event can be longer or shorter, can make as a consequence some different things. Like I like of timing. So if time is changing between the perspective that we are looking to, that then all the events that can are linked to neuron activity can have a different timing, and so this is that create like different brain activity, and also like networks that can be altered, for example, in pathologies where there is like a strange perception, for example, or something that we are not used to, and it's different also, it's difficult also to detect that there is not that connectivity, because also usually you measure that connectivity in some pathologies. So yes, I arrived in this ending. This was my interpretation.
Thank you. It's very interesting. Certainly, we have to do more in terms of understanding here. I was also thinking whether the prefrontal cortex and the higher form of interpretation of space-time is exaggerated in the in those frontal region where we tend to surpass the limitation of what the perception is allowing us at present. For example, if we cannot perceive the fourth dimension or more than that, right? Which is indicated in physics, but we certainly can derive mathematical interpretation and the possibilities that mathematics would allow us. So using those tools, or in terms of how probably Einstein did in terms of combining space-time dimension, where limitation of physical perception was surpassed by imaginative processes, where we could merge things in space and time, which is not really possible by the physical capabilities in the brain.
So yes, we are going to like more dimensions. So it's also in the paper it discussed about this hyperspace of events. So yes, we are simulating what is possible, what is not, if a connection between two events, so activation is possible in this framework or is impossible. So we are going like, as you said, like upper in dimensions. So we are trying to explain something that we don't can directly measure, but maybe if, as in the relativistic history, like from an idea, then they try to verify this indirectly, and so maybe also like at the end of the paper they discussed that maybe with like higher field some measures could be possible to also explain these findings or these hypotheses. So yes, it's a completely like new view that I never think about actually, and it was yes, interesting. And I think that also the frontal lobes maybe can play a key role in this, because correct me if I'm wrong, but maybe they are more in this conscious brain activities. So it's like a sum up, so something that we perceive as on the top of something that is happening behind in the network connection, and is like a result of all these events, maybe in these casual activations. So maybe why the frontal lobes is important in this framework.
Thank you very much. Thank you.
You're welcome.
Thank you both. Leah, just a quick answer if possible. There's a question on YouTube whether there is a relationship with episodic memory.
Okay, relations. So in the paper, maybe there were like also at the end a lot of examples. So maybe I missed that, but there was only this example of déjà vu, but I don't know if I can describe that as episodic memory. or no, it's not this. I haven't read maybe I missed that. I never had an application of these connections like in some pathologies that can affect memory or episodic memory. Don't know, but it's interesting because, of course, maybe it's an information that is there, but we don't relate to that, or there could be also like a space-time disconnection problem, but I don't know.
Or if you want to discuss it in more detail, feel free to move to YouTube. You can see the comment over there. Right, thank you very much. Michelle, are you ready for the next one? Let's unmute him. Here we go.
Thank you. Thank you. Still having a problem to share my thing. So it doesn't appear in the list of things to share. Got it.
Guess you're ready.
Great. So I'm going to discuss today this paper that really dragged my attention. Can you see well? Or
I can see a white screen.
You don't see the paper?
No, just a white screen.
No.
Looks like you shared a whiteboard.
Share. How about now?
Good.
Yeah?
Yeah.
All right. So this paper really dragged my attention because I've been really interested into the workspace. Actually, I had a grant about that. And the multi-domain network, which are like places in the brain that can get activated whatever you're doing somehow. So some people call it like multi-domain network, like a group of neurons that can be recruited to do anything and help you to have enough brain power to achieve what you want to do. And some others think that this is seen or the place in the brain where consciousness happens, and that would be in link with a global workspace. So if somewhere in the brain you have these places and neurons that have this superpower, the question is like, how does it happen? Where exactly will it happen? And the hypothesis mainly, which is tested in this paper, is that those regions should have more exchanges with other regions and within themselves to compute this higher power of processing information. So, and like originally, people were thinking that this was happening in the prefrontal cortices. That was all about, this kind of the prefrontal lobe is controlling hierarchically the rest of the brain. But then you had this parallel hypothesis from Bars who said that somewhere in the brain you have this global workspace that I just talked about, which would be linked to consciousness, like where you can stream what's happening around you through a loop that you can manipulate and modify, which is the fundamental requirement for consciousness, maintaining a representation and being able to change it. But the problem is like, those are theories, and it was not possible really to test it until now because we didn't have enough data. In order to test that, you need to have hours and hours of acquisition in the same subjects to test that. First, they have regions that activate whatever the function, and also look at the dynamic of the exchange between the region with resting state, as well as eventually with tractography, look at what happened if you cut some of the wires. But this is now possible thanks to the Human Connectome project and the big data that are freely available for anybody who want to access those big questions and not spend their life trying to get the money and acquiring the data before they can finally do the analysis and publish a paper. It's quite instantaneous. Although the authors, I had a quick exchange of email with the authors, said it took them 3 years to achieve this paper. So it was still like a long work. And so they really wanted to characterize the global workspace, where it is in the brain, And so the idea is like, if you have this global workspace, it should be at the top of the hierarchy, and it will be a core set of brain regions responsible for the integration and the orchestration of the functioning of the brain. And this, they measure it by a new statistical approach that will measure somehow the interaction between different brain regions that they call the functional rich club, the FRIC. Literally means money in French, so I found this very funny. And they use that using an information theory approach, which is a directed transfer entropy NTD framework that allows them to assess bidirectionally how the information goes in the brain. Does it get in the region? Does it get out of the regions? And do you have isolated networks that can process information in a loop and get modified by outside networks so you can update your loop and your representation, as I was saying. And then later on, you'll see that once they define the model, they also model virtually lesions inside this hierarchy and try to see what is going on. So that's a little bit of the idea. I think it's well represented here. The idea is that you have a hierarchy with a global workspace, which will be in the middle, which is highly interconnected within himself, but and receive information from functional modules that you can see here. That can be like auditory, sensory, and visual. They're getting inside the global workspace for this representation, get modified, and then get out. And they're trying to find where it is. And this is the result that they have. So they first did it with resting state functional connectivity. So they create these matrices that are bidirectional. And this allows them to quantify a little bit how much information are getting in all the regions and out of all the region. And you can see that for the in region is like mainly focused on the default mod network, and for the out regions it's mainly focused on the primary sensory and motor cortices, as well as visual. And then they have this other index, which is the total exchange of information that happen. This is highly correlated with myelin content in this region. So you can see here, the more you receive in or you send out, the higher your myelin content, which will be a representative of, like, you know, the process of sending a lot of informations. And then I try to see if this change when you're doing different tasks. And so what they're able to show here essentially is that compared to the resting condition, the information that is coming in change a lot. The profile of information coming in change a lot, while the profile of information coming out doesn't change that much compared to how much the profile of information coming in does. And which is an information that can give us some idea of how the brain works. So that's actually stated here, as it can be clearly seen what's coming on in the matrix, coming in the matrix, the rendering of the incoming flow of information are significantly different between task and rest. This suggests that different tasks process incoming flow differently. This is in contrast with what is coming out, the G out matrix, so the rendering of the outgoing flow of information, drivers, which are similar. This shows that sensory areas are consistently driving the information flow. Interestingly, it can be seen from the G total matrix that the total processing of information flow is more similar within the 7 tasks than when compared with rest, suggesting that the extrinsic sensory nature of task processing compared to the intrinsic nature of resting state processing. So your brain functions differently when you're doing things than when you're not doing anything. Then they try to look at like the areas that are in the intersection of all the frequencies. They find like those hotspots, and you can see here like 8 correspond to like the 8 different functions overlapping over there. And so essentially the idea, which is repeated here, is like you have this global workspace that maintained into loops or representations that can be modified according to value, focusing past, present, and also in order to decide what you want to do in the future. Last but not least, once they establish their model and the matrix, they've been able to compute prediction of what happened if you were to have a lesion inside or outside the frick. So the model, this is like the models that they created, which is a virtual model of the functioning of the brain, empirical measure compared to the model correlated at 0.6. And when they start doing a lesion inside the frick, you see that the correlation drop and the overall functioning of the brain seems to be impaired. When it's in not a frick region, then you can see that there is a significant difference between a lesion in the frick, but you still have a difference with the normal functioning. But their prediction will be lesion to the frick system will be the worst in terms of the overall functioning and the brain power, if you want, of individuals to interact and really being themselves. That's about it. There's a lot of supplementary material where they've been showing replication of their findings and the possibility to do that with other kind of parcellation, a lot of correlations, a lot of matrices. It is a beautiful paper. It's been very well achieved. I'm very impressed. Do you have any questions?
Thank you, Michelle. That looks like a very elegant paper. Any questions in the room? I see a lot of people.
Hey.
Patrick, yes, go ahead.
Really cool stuff. I'm just wondering, like the Rich Club has been defined before via means of graph theoretical approaches.
Mm-hmm.
Is there any overlap with existing literature?
Absolutely. So they use the Rich Club, and like they mentioned in the introduction, as a way to demonstrate their point and link it up with the global workspace and the variation of the interaction between what's not the global workspace and what's the global workspace in this frick kind of model, and how does it change. And what I forgot to mention is, like in the discussions, they link it up also with the gradient theory, because you can see that mainly the core of the frick is inside the principal gradient, and like, you know, like the most of the input is coming from motor cortices, and it's like this actually kind of color the gradient theory with a directional, like mechanistic functioning, as far as I understood. It's really good paper. I think I like really the perspective.
Another thing, when you said like the out parts of this frick and the in parts, basically, it kind of reminded me of like top down and bottom up processes. Think at that, like for a more theoretical embedding, even.
I think they do. I think they do overlap, although, like, you know, top down and bottom up is coming from the psychological, experimental psychology field, and here I think like the vision is more a directionality of the flow of information, but. And then they also study like this loop of what's going on within this network. Well, ultimately, yes, I can be linked up, but that was not in the introduction and the discussion. That's true. That could have been interesting to like try to expand it into that field. But it was already like a very well referenced.
Yeah, really cool. Thanks for presenting.
Are there any other questions? No. Yeah, Salvatore.
And thank you. And I was wondering, is there any analogy with the concept of brain gradients on the principal main gradients that I see in this figure, in figure 2? There may be a relation to the principal gradient described by Daniel Margulis and colleagues.
Yeah. Sorry, I was not clear enough about this. So it is in the discussion that the discussion with regard to the principal gradient. Let me find that. But mostly what they were saying is like it actually complements nicely the principal gradient, which are like just, we know those regions are correlated and anti-correlated. But here you have a directional kind of interaction between subregion of the gradient. So that gives really a mechanistic dimension beyond the gradient. I think it's here. Exactly. So a long history of neuroanatomy discovery has demonstrated that the brain is clearly hierarchical in its structure from single unit to the larger circuits. Research by Margulis and colleagues use neuroimaging to extend Marcel Mesulam seminal proposals that brain processing is shaped by a hierarchy of distinct unimodal areas to integrative transmodal areas. Recently, this idea has been further extended by applying the principle of harmonic nodes to the functional connectivity data. We extend this spanning using the advanced NDTE framework to establish a bidirectional information flow for identifying the functional hierarchical organization of the human brain. So beyond having like, the bidirectional onto your gradient, you get a high value, you get low values, you get middle values. Here you really have this direction of this talk to that in that direction. That's the add-on.
There is a lot of stuff going on here.
Yeah, it's going too fast.
Thank you.
Thank you.
Any other questions for Michel in his paper? Not that I can see. Great, thank you very much. Patrick, you have a paper for us as well.
Yeah, just go ahead and share my screen. A second. So, alrighty, it's kind of cool that we talked so much today about like embedding strategies, for instance, with the brain space or the brain space time, which is basically a 4-dimensional embedding, and now something that looks alike with Margulies gradients, which is also an embedding technique. And today I want to talk about this paper by Liang and colleague, which is in pre-proof and will be most likely published soon in Neuroimage. So it's about sex-related differences or asymmetries in the hemispheric functional gradient. Hence, it's right onto the topic of gradients. And what they basically did was, for the first time, to look into the differences in the principal gradient, like the principal and secondary functional gradient between the two hemispheres, so at the asymmetry, so to speak. And for this, they used the Aisha atlas, because one of the problems is to define areas that are homotopic in their functions. The Aisha atlas does this and defines 119 parcels per hemisphere, which are functionally somewhat equivalent. So when you don't take the whole brain, but the left hemisphere and the right hemisphere separately, and look onto the functional gradient by basically embedding the connectivity over time, or the functional connectivity across areas in a 1 or 2-dimensional space, you get the first and second gradient. You can use diffusion embedding for doing that, and you can embed as many gradients as makes sense. Typically, we're talking about the first 2 gradients or even only the first, the principal gradient, because it explains most of the variance. Now, when you do the same, not for the entire brain, but for each hemisphere separately, you're getting these images here on the left. So they did it in 700 something participants, only right-handed participants, with good contrasts from the HCP data set. And if you would compare this basically to the principal gradient by Magoulias from 2016, you would see that the gradients on both hemispheres quite resemble the principal gradient when you calculate it over the entire whole brain. However, for the second gradient, like they did it, sorry for that, they did it also in 2 different sessions. So 1 test session, 1 retest session, hence rest 1, rest 2, just to get a glimpse on the concordance between the two. So is the principal gradient or are the gradients stable in the same participant over time? That's true for the first principal gradient, but not for the second gradient when you separate the 2 hemispheres and calculate each gradient per hemisphere. So they just focused on the principal gradient, which looks, as I said, quite alike what Magoulias showed in his papers. So also, when you look at different functional networks, like the 7 networks defined by by Thomas Geo, then it's quite obvious that the default mode network is anchoring the gradient at one end, and visual and somatomotor cortices are anchoring the gradient at the other end, which is quite in line with theory about the gradients. Interestingly, there appears to be a sex difference in the asymmetry of these gradients. And that is when you look at the asymmetry of the full range of the principal gradient. So as you can see here above, the gradient scores in this case go from -2 to up to 2. And when you do this for both hemispheres separately and calculate left minus right or right minus left, you end up with this graphic showing a significant difference in this full range, in the variance or complexity of the gradient, with males having significantly higher full range in basically the left hemisphere compared to females. And that's true for the first principal gradient in both sessions. So it's a stable result. And also when you look into the areas that differentiate via basically an ANOVA, you can find, first of all, main effects of the hemisphere, as well as main effects of sex in various regions. So these areas differ in their full range, or in their, not full range, but principal gradient component scores between sex and between the 2 hemispheres. You see an hemisphere by sex interaction here in the middle part of the middle temporal gyrus, as well as the posterior orbitofrontal cortex. So this is the first time that someone looked into asymmetries of the principal gradients and also associated them with sex differences, which I found really intriguing and really interesting, because it opens up quite a lot of questions about, for instance, what drives these differences in the gradient, and what does it even mean to have like a bigger full range in the left hemisphere compared to the right hemisphere, which is modulated by sex. So there's still so much more to find out about these gradient representations of the brain. And I just think I cut it short here so that we can probably discuss a bit.
Thank you, Patrick. Very interesting. Just as a pre-question before I give the audience a chance to ask you questions, we had a lot of papers in Neuroccino already where we talked about sex differences and finding highly significant results. Are there effect sizes in your paper as well? Didn't see it when you scrolled through.
I need to check that. So obviously, I think the effect sizes again are very low. Just looking at the statistics right now. Let me share the screen again. Homotopic regions, rest, MTG, hemisphere by sex effect. We can see the effect sizes are minimal here, which is also, I think, already quietly nice visualized here, as you see, because basically these bar plots or box plots are so near to each other that when I wouldn't trust their statistic, I would say there's no difference. So I'm not really sure about it. If that's meaningful, that is. Like statistically, there appears to be this difference, but if it's a meaningful difference, that's on another page.
Cool. Thanks for the clarification. Are there questions? Victor.
Hey, thank you for the presentation of the paper. It was very interesting. I also have a question regarding the sex difference. Did they took into account the difference in brain size between participants and remove this confound? Because usually it can mean a lot of bias with the partial volume effect, and that can be found inside the later statistics.
So no, they did not compare for brain sizes. And I think the reason for that is basically it's, well, one could argue that brain size in the realm of functional imaging, where you define different parcels, basically doesn't mean that much. Then again, there's a counter argument, obviously, because of linkage processes, when you basically have the two brains or have brains of different size and merge them onto a standard brain, and in order to compare them, then obviously the smaller brain has to be stretched more. Hence the signals that you get from here that are transformed onto the new space are basically transformed more, or like the difference between the original native brain and what comes out after transformation is bigger than for a brain that already looks more alike the standard space. So what they did, though, was global signal regression, which should, in theory, somewhat compensate for this linkage issue. And I haven't looked at it in total because I don't think that they calculated their statistics, their sex differences, over the global signal regressed data. So I don't think they did it, because the only thing they showed here is how the principal gradient looks like derived from global signal regress data. But it might be in the supplementaries, and I might have just overlooked it. That's also quite a possibility here. So does this somewhat answer your question?
Yeah, okay, thank you, yeah.
Welcome.
Any other questions? Oh no, he was just waving at someone. I have a question in that case. So one of the figures, it looked like, here we go, the posterior middle temporal gyrus seems to be an important region. Is there an hypothesis in the paper why that region shows such an effect between hemispheric and sex interaction?
To be honest, I can't recall. So I haven't read the discussion, or can't recall the discussion entirely yet. I hope they did. In theory, I mean, when looking at something like volumetric asymmetries or something, we know that volumetric, and just going there because in volumetric asymmetries, you would see that the medial temporal gyrus is actually more rightward asymmetric than other regions, and which is differently in the planum temporale, which basically lies on top of the superior temporal gyrus. So, so the reason I'm mentioning that is that typically what we find when looking at like structural asymmetries, we see that males are sometimes like more stable in their result pattern than females. But that might be due to our bias, basically, because much of the studies we did so far, or older studies at least, sometimes only looked at male participants and disregarded female participants, or female participants in smaller sample sizes. So coming back to the question, I don't know if they discussed why this region exactly, or why the two regions exactly. But I'll find out in my next read through of the paper.
Lovely, thank you. And then a final question for me, unless there's another question. No, can't see one. So this gradient, I'm still struggling to wrap my head around it. So what does it, like, why do we care if it's more left or right lateralized?
Well, I mean, for me as an asymmetry researcher, I find it interesting to look at those things, because I mean the gradient itself, or the idea of representing functional areas not as distinct parcellated regions, but depict them in like in an approach that is capable of showing relations between these regions, where you basically have a two-dimensional plane, just for clarification or simplification, and put the dots, which now represent the parcels of all brain areas, next to each other if they are closer or more similar across their functional connectivity pattern, so across their connection to all other areas. This allows us to infer somewhat of the relationship between these brain areas, which cannot be done by simply looking at parcellation. So it's not that we're trying to map brain functions here, but really trying to understand the relationship between these brain areas across functions. And we know that the two hemispheres are asymmetric in their functions. So at least to some extent. There's obviously a relative asymmetry we're talking here. So for instance, with regards to speech or language, the left hemisphere typically is dominant for these kind of functional cognitive functions. The right hemisphere still does its part, right? So looking at this novel strategy or novel approach of interrelating regions to each other and showing that there may be asymmetries between the two hemispheres, even in this representation, is just giving ground to novel approaches of how to think about brain asymmetries or hemispheric differences. So I think it's quite interesting if you're into the fact or research about hemispheric differences, which is, and let me just clearly state that, which is like a very common observation, that we sometimes just see the brain as one, but our two hemispheres do things differently. And this is sometimes just neglected. So I think it's an interesting approach and interesting new finding to give rise to some more research in that regard. Because I also, just to sum it up, I also just thought that when you look at these hierarchical representations of brain functions, then the two hemispheres should be quite alike. Like, why should there be a difference? I mean, both hemispheres have somatosensory cortices or visual cortices. Both hemispheres have parts of the default mode network. So the anchors seem to be alike. Hence, why should there be even like a difference? But I never thought about like the full range as a metric of interest. So I think the real question here is, what does the full range even mean? Like, what is it related to? Anyway, sorry for branching out.
Thank you very much for that. Happily continue the discussion, but this is all we have time for today for Neuroccino. Thank you all for coming. Thanks for watching over on YouTube and for your questions. And we'll see you next Monday again. Bye.
Thanks a lot.
Thank you.